Welcome to today's presentation on post-transcriptional modifications. So, this is one of the very important area where we are going to discuss about how these modifications can be happen and what are the mechanisms which are involving, and finally how it is to be helpful to the final targets. So, in this section we are going to discuss about the rRNA and TRNA and mRNA modifications we are going to discuss. So, before we are going to discuss actually what is happening in post-transcriptional modifications, we will see the little basics that if you remember this slide, this is, we call them as central dogma of life. So, generally we can also call them as this is a central dogma of molecular biology. This was proposed by the Francis Crick, and where you can see here the information which is there in a DNA that is to be replicated or that is to be given to a next copy of DNA through the process is called replication. And the DNA, the information, whatever the information which is present on the genes that can be present on DNA, that will be given, that information is given to the RNA, that the process is, we can call them as an transcription. Then whatever the information which is received from DNA, this RNA has to translate it into a protein. So, that process we call them as an translation. So, in some cases the RNA, from RNA the DNA can also be possible to form. So, that process we can call them as a reverse transcription. So, especially this reverse transcription is we can see in retroviruses, but rest of all transcription translation will be basic to any of the cell. So, here our name suggests that post-transcriptional modifications. So, here the transcription means that is DNA to RNA, then modification after post-transcriptional. So, this is nothing but a transcription has done, then if it is entering before the translation there is a modification, that modification we are discussing under the post-transcriptional modifications. So, before we are discussing, we should understand two terms. What do you mean by polycistronic and monocistronic? We will start with the modifications with the messenger RNA. So, we are going to study or we are going to discuss here about three type of RNA modifications, that is messenger RNA, transfer RNA and ribosomal RNA. So, here we will start with an mRNA. On mRNA, that polycistronic mRNA, which is a very important criteria that can be existed in a prokaryotes. So, where one mRNA code for more than one polypeptide. So, one mRNA has come, then from the mRNA more than one protein or more than one polypeptide has coded, then we can call them as which is an polycistronic mRNA. So, monocistronic mRNA, this is an other sense, we can say it as this can code for only one polypeptide. This is one mRNA and one protein. This is very common or which is exclusive phenomena of the eukaryotic cell. So, here in prokaryotes there is no modifications can be done after transcription has happened, but if you see in eukaryotes there is a modification that can be happen before it is going for the translation procedure. So, if you see the diagram A here, we can see the green color DNA and the DNA where the red color is an mRNA. That mRNA, once it is to be translating, then the ribosomes has binded and the protein, the polypeptide has come out. So, this is called co-transcription has been taking place, co-transcription and translation in taking place in a prokaryotes. But if you see in diagram B, or that is in cartoon B, where you can find there is a special kind of structure that will be present where DNA is present, that we can call them as a nucleus, and this nucleus where exactly through the transcription the RNA has been started. So that is called mRNA has been synthesized. That we can indicate it in a red line. So after mRNA has synthesized, then which is going for the modification in the nucleus itself. So after modified or after modification has done to the mRNA, then it is to be transported into the cytosol, and where exactly the transcription translation has been taken place. So here the main important thing or main important remembering point is that post transcriptional modifications can happen only in a nucleus. So this should be very clear, and this is to be exclusive to the eukaryotes. So if you see this DNA, then on process of called transcription, then the RNA can be formed. This RNA generally we can call it as a primary transcript. For this RNA, from this RNA there are three different kinds of the RNAs has been formed. So we can say it as which is a tRNA, that is called transfer RNA, mRNA which is a messenger RNA, and rRNA that is a ribosomal RNA. So now we are going to discuss on this presentation how these three RNAs as done for the modifications after transcription. That is our main idea. So we will start with an mRNA. So mRNA which is a messenger RNA, that will be very important for the decoding of genetic information which is present on the DNA. So if you see the modifications about how this mRNA can taking place, because if you see that whatever the information which is present on the DNA, that can be given to the mRNA, and mRNA is the one which is like a messenger to the DNA or messenger to the genes. So this is a very important mRNA, that that process where three different kinds of the modification can be taking place for this mRNA. So before this mRNA is entering into the translation, it has to be go for the three kinds of the modifications. Those are, first one is we can call them as this is an capping. So the capping of mRNA, which is in first modification that has to be done at 5 prime of the messenger RNA. Then second modification we can call them as this is tailing. The tailing, this is done after completion of the or after complete synthesis of the mRNA. Then the modification, the tailing modification can done at the 3 prime end. So we can call them as this is called 3 prime tailing. Then the third modification which is very important, that is called splicing mechanism. So the splicing mechanism, which is a very important mechanism where we should understand that the gene arrangements, when gene arrangements in a eukaryotes when compared to the prokaryotes, there will be a major difference is there are non-coding sequences or non-protein coding elements can be present in a eukaryotic DNA, and that can be removed and only the coding parts can be joined. So that process we will learn how it is to be happen. That modification we will discuss in a splicing mechanism. So we will go with the first modification, that is called capping. So if you see this, the beta globin genomic DNA, where you can see the after the primary RNA, the RNA after synthesizing of the transcription, we can say it as this is an primary transcript or nascent RNA. So this nascent RNA, where exactly this modification can happen, there are three modifications which are very important as I discussed in a previous slide. One is capping. Capping in sense that is addition of 7-methyl guanosine cap at the 5 prime end of the mRNA. So this modification, especially this cap modification or addition of the 7-methyl guanosine cap at the 5 prime side, we can call them as this is the first kind of the modification to the mRNA. Then the second modification here we can see that is at the 3 prime end, where exactly we can call them as poly A tail addition. So, this is nothing but addition of adenine nucleotides to the end of the 3' side of mRNA. So, the third modification is which is a splicing where the non-coding sequences can be removed. So, if you see in this example, we can understand how the genome will have the blue color one which is a non-coding sequences and red color one the coding sequences. You can see total 147 base sequences can be present, and will removing everything, will remove the non-coding sequences, and finally you will get the coding sequence of that beta globulin mRNA. So, this is ready for the translation, and finally it can produce the protein beta globulin. Then eukaryotic mRNA is inherently unstable at the end. This is very important. Why this modification has to be done to the mRNAs? So, especially that messenger RNAs. So, the idea of this making the modification to the mRNA is to be making a very stable and which is to be having an half-life should be and more. So, in this criteria this modification can be done. So, if you see here the capping reaction begins soon after transcription has begun. So, once the transcription has begun, then the capping reaction has started. This is called co-transcriptional modifications. So, this will start at around the synthesis of 20 to 30 nucleotides are formed. Then that is the pre-mRNA, we can call them as, or we can call them as, this is a nascent mRNA will undergoes transcription. So, on this transcription there is a mechanism which can explain how this transcription can be taking place. So, here you can see here at the 5' end, definitely that is to be nucleotide triphosphate can be present. So, nitrogen bases, which are nitrogen bases we can see here, and adenine, guanine, cytosine and thymine, but this is an mRNA. So, here instead of thymine uracil can be present. So, here if you see at the 5' end 3 phosphates, that will be triphosphate, we can label them as an alpha, beta and gamma. So, there is an enzyme, that enzyme we call them as phosphatidylate, that enzyme you can call them as phosphohydrolase. That phosphohydrolase which can break the bond between beta and gamma. So, where the pyrophosphate can be released. Then the existing is nitrogen base with the diphosphates. So, on this there is a transfer of guanosine. So, that is from the GTP, we can call them as guanonyl transferase. This is an enzyme which can take the guanosine and that can be attaching to the 5' side. So, where exactly this addition is, you can see at my right side diagram, this is 5' to 5' linkage. So, 5' to 5' linkage, which is a very unusual linkage that can be taking place. So, this whenever the nucleases that can be present in the cell, those nucleases will not to be recognized this 5' to 5' linkage and will not have any kind of the action on this kind of the bonding. So, after this the methyl group can be transferred at the 7th position of guanosine, that is called guanosine 7 methyl transferase. Then where exactly the methyl group can be presented at the 7th position of guanosine. You can see at my right side at the top, at the 7th position we will see the methyl group. Then after that there is a methyl group addition can also be taking place at the second, the pentose sugar at the 2nd position of oxygen, where methyl group again be added. So, this is, you just remember it after capping mechanism. So, what do you mean by capping? The capping is nothing but which is an addition of 7 methyl guanosine at the 5' end. That is the first point. And the second important point is which is to be unusual nitrogen base pairing. That means whenever the nitrogen bases are pairing, that is to be 5' to 3'. So here this is 5' to 5' linkage can be taking place. This is called phosphodiester linkage. So this is always to be unusual. So this kind of the mechanism which can be operated whenever the mRNA is synthesizing. So this is called capping mechanism. So what are the advantages of capping mechanism? What are the advantages of capping mechanism after done to this mRNA? So we can give an advantages in a 4 different manner. First one, it can give the protection from degradation from ribonucleases. So you can just now we discussed about the unusual base pairing, that is called 5' to 5' base pairing. This 5' to 5' base pairing which is always to be an unusual base pairing. So ribonucleases which will not going to be have any kind of action, or which will not going to be have any kind of breaking actions on these unusual base pairings. So that is the first advantage. And the second advantage we can say it as which is an stabilization of mRNA. So this modification will give the stabilization, that is called which can improve the half-life of that mRNA. So the third important point is called which is enhanced splicing and translation. So this is very important. Once it is to be done, the modification has done, that is to be enhance the next level of modifications in a splicing, as well as which is very important for the translation. Then last fourth one is called enhanced transport from the nucleus to the cytoplasm. As my previous slide we discussed about, this modification can be happen in the nucleus itself. So once this modification has done, then this mRNA, the modified mRNA, or we can call them as which is a matured mRNA, that can be transported from the nucleus to the cytoplasm. For this transportation purpose also this cap structure will be very important. Right? Have you understood this? Now we are going for the second kind of the messenger RNA modification, that is called polyadenylation complex. So generally now we are discussing at the 5' end modification. Now we will go with the discuss with the 3' end modification. On 3' end modification we can say it as this is called polyadenylation. So the polyadenylation, if you see here, we can find the polyadenylation signature sequences in a human genome or most of the animal genomes. So that signature sequence we can call them as, here you can see here 2 uracil, 2 adenines, 1 uracil and 3 adenines. So these signature sequences, whenever it is to be expressing in a mRNA, then that is an indication that now the process has to be stopped and now it has to be initiated for the polyadenylation process. So this signature sequence around 10 to 30 nucleotide base pairs we can find, on 10 nucleotide base pair we can find the one more sequence that is called CA, cytosine and adenine, and from there to the 30 nucleotides we will see the GU rich, guanine and uracil rich sequences, or in some cases which is uracil rich sequences. So exactly what will happen here, the breaking of new mRNA can be taking place at the end of the CA, and where exactly the addition of polyadenosine can be taking place, which is up to the 250 molecules of polyadenosine base can be added to the end of the 3' mRNA. So this process is called polyadenylation. We can see here the polyadenylation, which is a little more complex mechanism when compared with this capping mechanism, where we should understand the few enzymes which are involving for this process. The first one is called CPSF. We can call them as which is an cleavage and polyadenylation specificity factor. This is a specificity factor that can bind to the signature polyadenylation signature sequence. That signature sequence is nothing but AAUAA sequence. So, this can be at the end, or this can be expressed towards the 5' side. Then there will be a second kind of the enzyme that can be present, that we can call it as CSTF. This is called cleavage stimulatory factor F, which interacts with the downstream GU sequences, or which are located at the 30 nucleotides below the or downstream of the signature sequence of polyadenylation sequence. So, where it can be bound with this GU rich sequences and also will make the contact with the CPSF. Then there will be two enzymes that can also be attached with this same complex, that we can call them as CF1 and CF2. This is called cleavage factor 1 and cleavage factor 2. Actually, what is happening here? If you see the left side of the diagram, you can understand. So, whenever the mRNA is synthesizing, then the mRNA where the signature sequence can be presented, that signature sequence, once it is to be exposed or once it is to be synthesized, immediately CPSF can bind to that signature sequence, and binding of this CPSF will make the binding of CSTF, and that will be more distance, that is to be at GU rich sequences, and the region which is there in between this CPSF and GU rich sequences, where exactly the CA can be present, that particular place will form the loop like structure, and where exactly the CF1 and CF2 can be bind. Then after this, the cleavage has been taking place. Whatever the unnecessary or whatever the sequence that is to be present in between the CPSF and the CSTF, that thing can be removed with the help of cleavage factor 1 and cleavage factor 2. For after removing of this, then there is an enzyme that is to be involved here, that is called PAP, poly A polymerase, which can cleaves at an poly A site, because where in between we will find the CA sequence, and after A this PAP enzyme or poly A polymerase enzyme that can cleave after A, and the cleaving will make you to be addition of the new poly A tails to that A. So, that can be taking place with the help of PAP2, which we can call it as, which is in binding poly A binding protein 2, and PAP2, which is in very accelerates the addition of A or adenine to the PAP. So, after this process has been this process has been done, and finally this can be continued up to 200 to 250 residues have been added. So, at the end, so after adding of 200 to 250 residues, then that process of termination can be taking place. There is a stop signals that can be expressed after this, and in a special kind of a protein that can be recognized, that we can call it as called PAP2, that will be recognized the stop signal, and finally the addition of the poly A molecule can be stopped. So, here you just remember this whole process is called tailing mechanism. In generally we can call them as, which is an tailing process. So, on tailing process, simple, I will tell you within 3 lines. So, on tailing process, this can be happen at the 3' end of the mRNA, right. Then the second one is on tailing mechanism, whatever the sequences which are to be important for the division. So, that process can be taken place. So, here finally what will happen, the addition of 200 to 250 residues can be taken place, or the addition can be up to 200 to 250 adenine residues can be added at the 3' end with the help of 3 enzymes. We should remember, first one is called CSTF, second thing is called CPSF, third thing is called PAP. So this is called polyadenylation, or this process we can say it as, which is called tailing mechanism. Then we are going to discuss about the next process. So this is, so far this is the second modification which has to be done for the mRNA. Now the third modification, which is to be very important modification, that can be taking place for the this messenger RNA, that we are going to discuss now. So before we are entering to that, I will discuss about two important points why this tail has to be added to the mRNA. One thing is the poly A tail makes the DNA molecule more stable and prevents its degradation, right? Then that is the first advantage. And what is the second advantage? The second advantage is poly A tail allows the mature RNA molecules to be exported from the nucleus. This is also very important. The nuclear export mechanism that can be done after this capping and tailing is completed only. That is a very important, we should understand. These are the few important functions for the addition of the poly A tail. Then we are going to discuss about the third modification of the messenger RNA. This third modification of the messenger RNA, we can call them as, this is called splicing. So on splicing, generally we should understand this is very exclusive, or all these modifications are very exclusive to the eukaryotic or eukaryotic cells. So here if you see, the mRNA is called, the mRNA which is present on eukaryotes, we can call them as, which is hnRNA, that is called heterogeneous nuclear RNA. So why we are calling as heterogeneous nuclear RNA? Because if you see the structure of DNA, there will be coding sequences will be present and non-coding sequences can be present. So here the coding sequences, we generally call them as exons, right? What do you say? We will say about the coding sequence is nothing but that is an exon. The exon in other term we can say it as, which is an expressed sequences. So which is a segment of DNA that is to be transcribed. So what does it mean? So whatever these exons can present on mRNA, finally it is to be coded to the protein, it is to be coded to the amino acid, and finally it will make the protein. That particular part we can say it as, which is an exon. The second type of sequence that can be present in eukaryotic DNA is called introns. Introns are non-coding sequences, or we can also call it as intervening sequences. What do you mean by intervening sequences? Intervening sequences mean these introns can be present in between of exons. So these introns is any segment of an interrupted gene that is presented in a matured RNA product. So here whatever the matured mRNA, if you see the mRNA, then previous modifications are before the post-transcriptional modification, we can say it as, that is to be a nascent, and after modification we can say it as, which would an matured RNA. So on matured RNA, these introns can be removed and only exons can be taking place. That is very important. So that process we can generally explain in a mechanism that is called splicing mechanism. So before you are going into see how exactly the splicing is taking place, for that we need to understand few basics. So we can see here the cartoon, you can see there are three areas which are very important. So the area which is there in middle, so that is to be written an intron, right? Then at my left side and right side we can call them as, this is exon 1 and exon 2. So here there are 2 cuts or 2 splicing sites can be present on this mRNAs. So, one towards the 5 prime, we can say it as, which is in 5 prime splicing site, then second splicing site at the 3 prime. Then in between, if you see the intron, in between there will be a special kind of point can be present, or special kind of bases can be present. We can call them as, that is, a branch point. So, the branch point is here, which is always to be uracil or cytosine. You should remember that the branch point is always can be present in only introns. So, you clear, right? At the 5 prime site, at the 5 prime side, the splicing site can taking place, and again one more splicing site at the 3 prime side, and in between there is a branch point in a intron. The branch point will have the nitrogen base, that is always uracil or cytosine. Clear? So, if you see, generally this we can say it as, which is GU-AG rule. So, this particular rule describes the presence of these constant dinucleotides at the first 2 and last 2 positions of the introns of nuclear genes. Then the second one is splicing sites that can be present, and there will be after splicing. So, what will happen? The intron part can be removed and the exon part can be joined. That is called splicing junctions. This process can be happen at the splicing junctions. So, we will go and see in detail how this can be happen. So, here I have given a few one important gene, and this can code for the ovalbumin, which will have the 7700 base pairs. So, after transcription and then capping polyadenylation mechanism, then after splicing, now we can call them as, this is a matured ovalbumin producing mRNA that will have only 1872 nucleotides. You just imagine where this whole gene will have the 7702, the expressed part is only up to 1872. So, where you just remember it, where you see the light orange color that can indicates the introns, you can see the introns, total 7 introns can be present and the total 7 exons can be present. So, as the exons are nothing but these are the coding areas or coding sequences, this has to be come to the matured mRNA. So, if you see down the last one where only the exons can be, the joining of exons can be taking place, but not to the introns, that can be removed. So, this whole process will ready, and that matured mRNA now it is ready for the translation mechanism, and finally it can produce the ovalbumin gene. So, if you see this, the splicing which is a very important phenomena, and this can remove the non-coding sequences. So, this splicing generally will be done at the special kind, that is nothing but post spliceosome mediated splicing, or generally we can call them as spliceosomes. So, spliceosomes are a special kind of the complexes where this splicing procedure can happen. The spliceosomes contains a specific set of base, that is uracil rich snRNPs, that we can call them as small nuclear RNPs. So, here if you see the complex of the spliceosome will have the RNA will combined with a protein, we can call it as small nuclear RNAs, which are very important for forming of this spliceosome. On this spliceosome only the introns can be removed. So, if you see that process how which is to be done. So, generally you can see here we discussed as the previous slides where the splicing site can be present, that is called recognizing site, and in between the branching site can be present, that is in the intron, and finally the catalyzing site can be present, which can where it can be cleaved and will be joined with the exons. So, if you see the arrangement of the spliceosome, on spliceosomes there are molecules, generally we can say it as which is in protein and RNA molecules, right. The protein and RNA molecules, these are to be on various kinds. So, this is like U1, U2, U4, U5, U6 and so on. So, these are, if you see the function of this protein, the U1 is nothing but which is a combination of RNA as well as protein. The U1 will recognize the 5 prime site recognition. This is very important. So, if you see, if there is a sequence, so where exactly what will happen at the 5 prime site, there is an 2 splicing sites can be present. 2 splicing sites can be present at the 5 prime site, that can be recognized by the U1, and U2 can be recognized at the branch point, and U4 can be again which can pair with the U6, and U5 with the pair with the U4 and U6, and finally the arrangement will be like this, you can see in this picture. So, if you see the mechanism, how exactly it is to be operated. So, the middle line which is to be an intron, the red lines to be an exons. We can see that the splicing site will be recognized by the U1, then the branch point A, that can be recognized by the U2 and will form the loop like structure, and this loop is to be finally the loop can be removed. You can see the loop can be removed and the exon and exons can be attached. You can see the final product which is to be an exon 1 and exon 2, the red color one, these two things are attached, and the introns will degrade, and the removed introns can degrade, and again the SNRMPs are used again for the next splicing mechanism. So, this is how the splicing can be taking place. So, remember this can be done at the spliceosome mediated complex, and where with the help of the U1, U2, U4, U5, U6, and where these complexes we can call them as proteins along with an RNAs. So, there are a number of theories that can explain how this introns can be removed. The next kind of the mechanism we can call them as alternative splicing. So, alternative splicing which can be firstly identified in viruses, later on that can also be identified in eukaryotes, which is first discovered at 1970s. So, the first alternative splicing mechanism has been reported in IgM, immunoglobulin M gene. So, where you can understand what is splicing and what is alternative splicing. So, here the RNA splicing, generally what we discussed in a last slide, that is called splicing. So, there is a difference between alternative splicing and splicing. The splicing is the process of splicing the exons of the primary transcript of mRNA, where the alternative splicing is. So, that is nothing but the only addition of the exons, that is called splicing, but in an alternative splicing the process producing different combinations of exons can be attached. So, here if you see, I'll explain in this slide. So, if you see this slide, you can see the total exons can be 5 and introns can be, you can see there. But in my left side you just understood that after maturation what will happen, the combination of 1, 2, 4, 5 exons can be joined. So, here the exon 3 can be skipped. So, that will give you a special kind of protein that we can call it as, which is a protein isoform 1. So, that will have a special kind of function which is called function A. But if you see my right side, where exactly what will happen here, exon 4 is skipped. So, here 1, 2, 3, 5 come. So, this will have a different kind of the protein sequences and will have a different kinds of the function. So, this is a very important phenomena where that can same mRNA can produce different kinds of the proteins that are very important for the splicing. So, finally, what will happen after the matured mRNA, capping starts, coding and poly A tail can be happen. So, if you see the TRNA generally 4 different kinds of the modification can be present. One is to be removal of the leader sequence that can be present at the left side, and replacement of the nucleotides that is to be with the unusual things, and excision of the introns can be taking place. You can see here the first one, the whatever the cleavage that can be done and modification of bases can be happen, that is with the pseudo uracil, 1-methyl adenine, 7-methyl guanine and 3-methyl cytosine. These are the different kinds of the modification of nitrogen bases that can be happen in the tRNAs. So, that is a little modification which is related with the tRNA. Now, the ribosomal RNA will have the modifications. In generally, you can see the ribosomes where 50S, 30S or 60S, 40S can be present. So, if you see the ribosomal modification, we can understand. So, the first process of the synthesis of the ribosomal mRNA, that is to be 45S molecule. So, where the first cleavage can be taking place, that can give you the 41S intermediate plus 18S rRNAs. Then on second cleavage, this can be taking place at the 41, that can give you to 32 intermediate. So, on the final 32 intermediate, finally it can give you the 28S and 5.8S. So, altogether 28S, then 5.8S and 18S RNAs can become with the same of the 45S molecule. You just observe here. So, that is called transcription and 18S. These are the different kinds of the RNAs that can be present in a ribosomal RNAs. This will explain you in this diagram. You can understand that first which is come all kind of the RNAs will only in a 45S manner, then after that it will come the first RNA will come is 18S, then later on and 28S and 5.8S. So, if you see there is a special kind of RNA that can be present in ribosomal RNA, that is called 5S RNA, but this 5S RNA that is not going to be present, or this sequence will not to be present on that 45S molecule. So, generally if you observe this can be coding by a special kind of RNA polymerase molecule, that is called polymerase 3. So, this will not have any kind of the relation with the coding of 28S, 5.8S and 18S RNAs. This rDNA, the DNA which is having the 5S RNAs is present outside nuclear organizer. So, generally what will happen, this can be code with the help of the RNA polymerase 3, and that can be present, or this particular part can be organized in a nucleolar region. And for this transcription purpose, there are two proteins, transcription factor 3A, B, C, these are to be important. So, these are certain modification which will be taking place for the ribosomal RNA. So, now coming to the conclusion that now so far we are discussing about the post-transcriptional modifications. On post-transcriptional modifications, we are going to discuss about 3 different kinds of the modifications. So, especially 3 different RNAs modifications. One is mRNA modification, second thing is tRNA modification, third thing is rRNA modification. So, where on mRNA modification which is to be highly studied and well identified, that is to be 3 kinds of the modification. So, that part we generally call them, or we discussed in capping, tailing and splicing mechanism. On tRNA there will be unusual modification, there will be next time will be the final can be the leader sequence can be modified, and the final the introns can be modified in a tRNA. But if you see the ribosomal RNA, where the ribosomal RNA that can come with the same RNA, that is to be 45S, from there to be 28S, 18S and 5.8S can be formed. Then finally, the synthesis can be taking place, the 5S RNA can be taking place at separate, and this can be organized in nucleolar region with the ribosomes. So, with this presentation, I hope you can understand the post-transcriptional modifications, and thank you.